Reliable Automation with Human Oversight. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who have registered on xtalks.com. At this point, I'd like to thank Bullfrog AI, who developed the content for this presentation. Bullfrog AI leverages artificial intelligence and machine learning to advance drug discovery and development. Through collaborations with leading research institutions, Bullfrog AI uses causal AI in combination with its proprietary bfLEAPTM platform to analyze complex biological data, aiming to streamline therapeutics development and reduce failure rates in clinical trials. Now I would like to introduce our speaker for today's event. Juan Felipe Beltran is Director of AI Machine Learning and Innovation at Bullfrog AI, where he builds explainable ML platforms for clinical trials and drug discovery. He specializes in human-in-the-loop systems that transform complex biomedical data into production-grade analytics, with experience in processing thousands of clinical samples from brain banks and multi-omics studies. But now, without further ado, I'd like to go ahead and hand the presentation over to our speaker for today. Juan Felipe Beltran, you may begin when you're ready.
There we go. Good morning, folks. Today we will be covering clinical data analysis with agents, looking at reliable automation with human oversight. Before we begin, I wanted to give you a little bit of information about me. I am a computer scientist by training with a PhD in computational biology. During my time as a graduate student, I worked in many problems on protein-protein interactions and how they relate to health, how genetic mutations can break these systems, and how we can use those insights to find better ways to target disease. After that, I did a postdoc where I expanded this work into the human microbiome, went on to work at Quantum Sci, where we designed the world's first real-time benchtop protein sequencer, which had some very interesting data problems related to the work I'm going to be talking about today. Afterwards, I worked at Parita Sciences, where we were looking more of a biological applications focus. How do we take diverse genomes like those of bats and mine them for insights that might let us know what they are doing differently biologically that allows them to be so resilient to some of the diseases that we struggle with? Now I work at Bullfrog AI, where I have the privilege of working with some pretty amazing code bases. We have an exclusive partnership with the Applied Physics Lab that allows us to use a lot of algorithms that are usually used for outlier discovery in national security applications for biological data sets, so that we can connect applied physics, statistical machine learning, and LLM-in-the-loop algorithms to systems biology. We also have some very interesting data sets that we've analyzed, like 2,800 brain samples, which had RNA sequencing, and we used to look at schizophrenia, major depressive disorder, and bipolar disorder. Across all of these fields, the question that I get is always some form of, can you find a pattern in the data? And it's the question that we most often are asking in biotech. Pattern can be a treatment-resistant cohort, some therapy escalation trajectory, or risk scores. They can be new drugs, they can be anomalies in the data. We answer all of these questions as part of a service that we offer through bfLEAP, where we find similarity networks and dependence networks to answer these very difficult questions. But there's a question that gets asked immediately before we're able to find any patterns. How do we make pattern finding possible for our tools? Now, we're going to be looking at 3 different ways in which we make pattern finding possible that might get skipped depending on the type of work that you're familiar with doing. First, we'll be looking at building relevant feature combinations from a data set, building relevant categorical features in a data set, and then structuring trapped data. I will keep this talk approachable to people without a lot of technical experience in these areas, particularly hitting at the impact of why we want to do any of this. But I'll include enough technical detail that if you are used to coding, if you're scripting things and sending them to any of the LLMs that are available right now, you'll come away from this with real takeaways that can improve the way you're doing your work. Throughout the talk, I'm going to be focusing not on how to do this with AI, that is a losing proposition, but rather how and where do we incorporate AI into our workflows where we're trying to achieve these goals. So to begin, we're going to look at building relevant feature combinations. The punchline here is fairly straightforward. Your model is never going to be able to guess what a domain expert knows. If you have some data set that has age, height, weight, sex, glucose, temperature, and BP, and you're trying to predict some form of risk for your data set, you are not going to be able to force the model to realize that height and weight can be combined into BMI. This is especially true if you've normalized these features in any way. What does this mean? The model can find that there's some complex relationship between height and weight that might affect risk, but it's not going to be interpretable. And if it's not able to find this complex relationship, it's going to hurt your performance at the end of the line. So how do we look at this? What does this look like in actual data? Usually, we have some clinically meaningful features that we've extracted from a data set. Here, I'm giving an illustrative example with 3 patients where we have their age, their sex, height and weight, creatinine, white blood count, and neutrophil percentage. And if you work with any data sets like these, it's very common to just put them into your machine learning model or statistical analyses if you don't have domain knowledge on board. But there's features that are missing here that are derived from the features that you're looking at. Not only do we have BMI, where we take weight over height, which can be a very powerful measurement for obesity or nutritional status, but we have more complex features. eGFR is a function that you fit on creatinine, age, sex, and race. And it's a feature you're going to want to have in your data set if you're interested in kidney function. ANC is the white blood count percentage that's not coming from neutrophils, which is very useful for infection risk. And CRCL is a function of age, weight, sex, and creatinine as well. You want to generate all of these features. Any clinician could tell you that these features exist, but without domain knowledge, you're unable to do anything with them. So how do we solve this? How do we know how to build these features? Which features do we even need to build? Option 1 is to find a domain expert that's not busy, which is going to be very difficult, thinks in ML feature terms, which is rare, but it's getting more common, and loves open-ended questions, which is, I think, the most frustrating part about trying to do this with a domain knowledge expert. You can't just bring the data set to them and ask, what features should I be adding to this? What do you think? That tends to be a worthwhile thing to do once in a while, but frustrating on their end, especially if you're analyzing a lot of data. Option 2 is to scour the literature, knowing that it's going to be scattered across decades. It's going to be therapeutic area specific, and it's constantly evolving. The insights that you're going to find during your dive are going to come at a high cost in terms of how much you look, and the reward might be something that you don't actually want to use at the end of the day. Option 3 is one of my favorite ones, regardless of what the problem is, which is to find the open source helpers. No matter what problem you're looking at and no matter what field, you will find that there is a small group of people that are working on building tools that everyone can use. You can find out what their tools and resources are. In this case, Hades has a phenotype library and feature extraction library that do this type of engineering of certain features. But inevitably, you're going to have to understand what their format standards are and translate your data into their data models. OMOP is a really good example of a common data model that is powerful, but can be a big lift to incorporate into. OMOP is basically a database structure where you have specific names and concepts that you're going to apply to each of the measurements that you have in your data set. You have tables for each person, for condition occurrences, drug exposure, what have you. More importantly, I think you have standardized vocabularies. Your conditions are going to be in some SNOMED identifier, your drugs in some RxNorm identifier, your measurements in some LOINC identifier. Getting your data ready to upload into OMOP is a major lift, but you want to do this as a way to browse a feature menu. There is structure and ecosystem that becomes accessible when you look at putting your data in an OMOP common data model. that would allow you to go in and fish for what features might be relevant in this area. This can be done through their forums, through open source tools that already exist, like the ones I mentioned before, modules in Python, modules in R. That being said, tool coverage will depend on community contributions, as they always do. That is the Achilles heel of looking for the open source community. To give you an example of what's difficult about this and how we accelerate it, let's talk about entity resolution. Entity resolution basically comes down to you have some term in your data set. You have a column that says for one of the rows, hypertension, comma, uncontrolled. And your task is going to match that to an OMOP concept ID. You can look through the OMOP concept IDs and you'll find that each of them is an identifier that is linked to some concept like hypertensive disorder or hypertensive heart disease. Already here you can see that it might be a little complicated to match an ID to its concept ID or a string to a concept ID if you don't have a perfect match between what your data says and what it says in the database. You can do this manually, especially making sure that you're making decisions that they would be made by a domain expert. You can use fuzzy string matching, but it tends to be a pretty poor choice in this area. You basically script it to find similar types of text. And whatever you do, you're going to have to make sure that you keep pretty good notes because you're going to be repeating this for thousands of terms and want to make sure that you know how each decision was made. This is where we see a fantastic use for the AI agents that are available now that can use some amount of reasoning, some amount of web search, and some amount of guidance. The general framework that you start with is having some item that you're interested in. Here I represent it as a ticket that you're going to be submitting to an agent prompt template. So you don't want to write a full prompt every time you're interacting with a language model. You probably have some fill in the blank style of prompt that you're going to drop hypertension, comma, uncontrolled into. In this case, if this is all we do with the system, we're going to get some wall of text from ChatGPT or Claude or Grok, whatever model you're using, and it's going to say something like, happy to help, here is what I think you should do. And it's going to have some caveats because we're getting better at having them not just give you an answer if there is not an answer. This is not super useful. You have not mapped any IDs. You may have cut your work down a little bit, but not much more than you would have with a quick Google. One of the things that we can add is context. Additional patient information, for example. This is something that you could add as a full data dump of what is all the patient information for this patient that has allegedly hypertension, comma, uncontrolled. But MCP is a protocol that allows you to give the agent an opening into some specific type of data or some specific type of task that they can perform on the computer you're running this from. If you're running your model in the same computer, which is preferable, they'll be able to look, let's say, at the table that you have of patients and look up some of their measurements. You can also use this to give them access to previous decisions on previous patients that had a similar item, or have a file with the preferred guidelines that you'd like to follow, which can be a combination of the regulatory guidelines plus any notes from your team. Here we can see that it changes the response significantly. It uses the tool to look up recent blood pressure measurements, medication exposures, and condition occurrences, and then applies a specific rule to it to decide what should happen to this term when it's mapped to a concept ID. This is still not quite useful. You're running this for thousands of items. You're not going to be able to read through all of these and make them actionable, make decisions, or triage them in an efficient way. And this is why you need two more gadgets to add to this. First, structured output. If you've heard of Pydantic, you know exactly what I'm talking about. If Pydantic as a concept doesn't make sense to you, this is simply something you can put at the end of any language model to make sure that the output follows a specific set of rules, which can be a structure like the one you're seeing on the right. This turns the task from something that you're doing in a chat interface that is giving you answers as a human to something that can be processed by a computer, that can be scripted around, that can be analyzed across all of your queries so that you can make decisions in a more informed way. You don't want to use this directly for decisions, though. As you can see, it's overwhelming. So the last step that we use in our processes is to do human review, to build a small interface in which you can have your data scientists and your domain experts make quick approve, reject, or refine decisions on whatever the recommendations are for the specific term. In this case, it shows you what the recommendation would be, what concept ID it would apply it to, and it can list evidence, assumptions, provenance, and raw JSON. What's nice about this is if the decision is trivial, you can take a quick look at this, press approve, have that logged somewhere, and have the provenance of the rest of your data tightly controlled. If this requires more iterations, you can field this as a refining task, or you can assign it to further triage. This general pattern is extremely useful, and it is what I'm going to be referring to throughout the talk when I say that we're using language models in a programmatic or systematic way. We're using prompt templates that are going to take some set of inputs repeatedly, some items. They're going to have some context available to them. The context is usually available across items. And they're going to output something that is structured so that we can build it into a data analysis pipeline or into a direct interface for human review. Now, this allows us to do mapping to the vocabularies that you need for OMOP very quickly, much more quickly than you would normally. We've cut that time by 90%. And we're very happy with how quickly this evolved as we started trying it. But the reason we went down this road in the first place is to do feature engineering, to figure out from our data, what are other features that I could be including or should be including before I start my statistics or machine learning pipelines. And here, depending on the field that you're in, the intervention that you're observing, your tool coverage is going to still depend on community contributions. So looking at what we've done so far, you might be thinking, well, why do you need to put this into OMOP at all or only if now you have a machine that can automate lookup and suggestions? And that's exactly what we do. We do de novo agent-assisted feature discovery. Rather than just relying on what the community has created, we can now use a different type of items, some of the columns in our data set, like ESR and CRP in this example. And we can add some context. The context can be very broad, like saying, this is a rheumatology data set. So make sure that you take that into account when you're suggesting features. And the goal of the analysis is treatment response prediction. So features that are relevant to that type of analysis are particularly important to include. You can also add an MCP hookup or a file that tells them what are other features that are available so that they can make better suggestions. Your output structure in this case is going to be making sure that you have formulas, citations, and clinical rationale. Clinical human review in this case becomes deciding whether or not you want to add it to the list of features that you're going to be using in your intake pipeline. Log CRP, for example, is something you would want to use. And if you haven't heard of it before, you'll have the citations available to you by following this output structure. Now, there's a pretty common question here that is, how do you know that these papers are real? Is it going to be hallucinating? And I know where these questions come from, but I'm happy to say that first, hallucination has gone down with every single release of language models that we have seen over the past year. But two, and more importantly, this is not a get out of doing work pass. This is not about automating an entire workflow using AI. What this lets you do is have new options when you're trying to do feature engineering. You have option 4, find a domain expert that is not busy, still difficult, but loves to argue, much easier. All they need to do in this scenario is shoot down a specific set of literature-sourced ideas in their field. This is something that goes much faster than asking them to de novo generate features that they think would be useful for your data set. Option 5 available to you now is to verify the literature, not just perform a literature dive. You have a curated shortlist, it's on a specific subject. This becomes basic due diligence so you can decide what to move forward with. And option 6, which is still useful, is you can onboard data to standard models like we showed in 10% of the time with minimal data reentry. The general framework here is we don't want to be writing prompts where we're asking ChatGPT to solve our problems. That is going to help in very specific situations, not when you are trying to set up pipelines that are repeatable and useful. What you want to make sure you're focusing on, and what we have been able to do at scale, is build prompt templates, structured outputs, and put this in a human feedback loop so that our domain experts and our data scientists can come together and understand what it is that we're generating from the data. The goal is not to automate, it's to responsibly scale using the best tools available. So that's how we build relevant feature combinations. This is all part of a larger service that we offered called bfPREP that helps you get to the start line of analysis when you have a data set that might be complicated or might be under annotated. Under annotation is something that we cover pretty thoroughly in the second section that I'm going to start now, building relevant categorical features. Now, there's a couple of problems when you're building categorical features that I'm going to go over. All of these are small data nightmares that you or your data scientist will have encountered at some point. First is the missing context problem. You have patients, you have medications that they have taken, their dose, route, but you're missing some information that would be very useful to whatever workflow you're using. So ideally, you would want to use some online database to populate mechanism, target, and class. If all you need is already available in databases that you have access to, like drug bank, then this becomes about mapping the medication name to a drug bank identifier, just like we would in the entity resolution example before. But there are some nightmares that are not as straightforward as ID mapping. The free text problem, which is extremely common with clinical data sets. You have some field that is free text that is filled in by different physicians. This can be in different countries, different institutes, different ways of teaching. And even for something as simple as the left ankle, you will find as many ways to say that as there are doctors. This becomes pretty useless as a data set, as you need a way to harmonize all of this into the same concept, ideally adding some new column that would say left ankle in a consistent way. But if you're looking at this and you're a doctor, you might be thinking, well, I don't know that if you're doing meta-analysis, I don't know that it matters that it's the left ankle. It might be just the ankle, or it might only matter if it's a particular leg, if you're doing some gait studies. And that is the third problem, the resolution problem. This is most salient when you have a data set with many different diagnoses, but the counts are very low. Now, these diagnoses are related to each other. You know that you should be able to group some of them into categories that would give you more power for statistical analyses or machine learning analyses. But doing this automatically is something that's not currently enabled. In this case, making a new category that's systemic CTD as a diagnosis group and idiopathic inflammatory myopathies as another diagnosis group. Here you can see we're looking at 43% or 40% versus 40% of the data set if you want to do a contrast between systemic CTD and other autoimmune versus any of the very low single-digit counts that you would have in the scenario on the left. So this is desirable. This is something we want. This is something I've wanted at many different jobs and many different data sets. The way that we would usually try to get this is by looking for ontologies that might be able to map our data. Some of these are open source, some of these are paid. They all look roughly the same. We have terms in a tree where the lower down you go in the tree, the more specific the concept is. And if you climb up a branch of the tree, you can go from systemic scleroderma to connective tissue disease. You climb up high enough in the disease ontology, you will find the most general term, disease as a whole. There's problems if you want to use this to try to solve the resolution problem. There's ID mapping, which thankfully we have a really good solution for now. But you still have to make a resolution choice. Which level do you collapse to? And it's going to depend on the data, what you're pursuing, and what the counts that are going to come out of that will be. This is something very difficult to do at scale and something very difficult to do without bothering a domain knowledge expert every couple of minutes as you look at different resolution choices that you could make for different labels. And finally, much like before, domain coverage is very sparse for some areas, even if it's well mapped for others. So if you're specializing in rare disease, these ontologies might not actually give you what you want. It might not be as easy as just taking the diagnosis. It might need more information about the patients, which is why, again, we can use this gadget where we can take a source ID or text and map it to a target ID to great effect. But we can go one step further and look at human-in-the-loop category design. This might look a little complicated, but it's a fairly straightforward process. We're going to ask the question in two layers. First, we're going to ask which categories make sense for a particular column, let's say diagnoses. And we're going to take subsets of our data set, just get the value counts of how many of each label we have for that subset of the data set, and send that to the agent to give a structured output. In this case, the structured output should be a list of categories that they are nominating. You can aggregate this over many different subsets, and you have the list of categories that you think are reasonable additions to the data set or reasonable groupings that you could use in the data set. Here you have a step of human review to approve the structure, make sure that you deny any superfluous categories that you don't think would actually add to the data set, and your result is a new structure. It's not a mapping, it's just what are the options that you think should be valid for this column. And then you run a separate workflow where you populate the column using that new structure as the output from the agent. This goes not to an interface for approval, as now you're populating all the rows in your data set with an additional data, but to a data scientist that can check how did this mapping occur, how high quality is the mapping, what are the counts of different ones, and are there any outliers or anything that looks off. So this is an example of trying to use not just human review, but making sure that your human review can come from the data scientists doing what they do best, finding patterns in the data. This pattern is very generalizable. It becomes find a schema, utilize the schema. And we use it for many of our applications that are sped up by language models or agents. In general, you can think of this as sampling from a data set, taking the structured output and either updating the context or exiting if you already have all the recommendations that make sense, or if there's nothing new to add from the sample of the data set. Then you have human approval, a new structure, and you apply the new structure when you utilize a schema to be reviewed by a data scientist. When we do this, we get categorical variables across the board. We get a lot of labels that allow us to describe patients in bins and batches and look at the covariance between these types of labels. We love categorical variables when we're doing analysis. The APL platform that I mentioned before does graph analytics particularly well with categorical variables, and we've used them in many different domains to great effect. So the second we saw that we could do this, we were very excited. We had a client that really needed to do this type of mapping for a clinical trial data set that they wanted to do post-hoc analysis on. And we were very excited to do this for them and find subgroups of patients that might have differential response, or issues between clinical sites that might have some signature of recruiting or dropping out of the study being anomalous, or adverse effects being anomalous. But when we engaged with this client, we found that we were going to have to develop a new capability, and that is structuring trapped data. And this is my favorite type of horror story with a happy ending. The data, we realized, was not a set of tables or even a set of scattered Excel sheets that are poorly formatted, the typical working grounds for bioinformaticians, but rather 10,000 plus pages of PDFs. This contained a variety of important data. Some of the pages had adverse event tables. Some of them had patient demographics. Some of them had lab results, medical history, screening forms, treatment outcomes. There was no way to dice these up from the beginning. And there was no way to get this data directly using classical methods. If you had asked me to do this a year and a half ago, I would have said it cannot be done. This is a trapped data set that's going to remain trapped. And I would have said that not because I think it's just a lot of work, but because the traditional approaches in this field have not generally worked for this type of data set. You can do optical character recognition to try to extract text, but we're looking at complex layouts, and complex layouts are not going to work well with OCR. There's tables that are spanning many different pages, so some of the information is fragmented across pages, and that makes it more challenging. There's handwritten annotations, and there's low resolution scans, some of them with a tilt. In general, OCR is going to have a very hard time, and you're going to end up running QC on each of these pages, if not each of the fields that you're extracting. The schema is also inconsistent. Each trial is going to use different table formats. Even different tables within the same trial are going to use different formats. Column names are going to vary. Units are going to be inconsistent, and there's going to be missing standardized identifiers. Sometimes we're going to have that free text problem. And the context is scattered. We have patient ID on one page, demographics on another, lab results on another, outcomes on another. And sometimes there's no clear linkage other than the descriptive statistics of the patient that could be mapped to some patient ID that could then be harmonized across the entire data set. And mixed data types and scale, as you can see on the right. It is a very difficult thing to do with traditional approaches. We were able to do this with a pipeline that ran, including human analysis, in a couple of weeks using a multi-stage human-in-the-loop pipeline powered by bfPREP. We have 5 stages to this pipeline. Each of these stages works as a different type of semi-automated task with a human review gate. Sometimes that human review gate is an interface. Sometimes it's a data scientist. Stage 1 is our per page triage. Classify and rate each page. What is it? Is it demographics, labs, adverse events? We were able to build into this even the difficulty of extraction with descriptions such as the difficulty of applying OCR to this page based on the schema, based on the quality of the scan. What were the most likely mistakes that you would make when trying to extract this? And we can take that triage as a data set and analyze it so we can find what are sets of pages that we can then verify as humans that have contiguous tables that we want to extract or statistical information we want to use later. Then we go to stage 2, which is the schema definition. As I said, you want to extract the structure before you start extracting the data. This becomes what fields will we need to extract information from each group of pages. If I give you the adverse events table, this becomes looking at what are the column names and are the rows patient specific, or does a patient have more than one row assigned to them? Once we have a schema definition, we actually extract the data page by page using the appropriate schema. This was a very exciting thing, not only to see work, but to see improve over time as different models were released. One very exciting thing about this became using the same exact pipeline to extract a data set we didn't think was something you could extract, and seeing very high quality when we did so. To be able to get this to a state where we were very happy with it, we had to learn a lot of tricks with preprocessing, but that is a subject for a whole other talk. As we found that the things that tend to help OCR systems are not the same things that are going to help an agent that's able to look at an image, extract data. Finally, we have cross-page linking as our last semi-automated step, where we link the records across pages to build complete patient profiles. And then we're back to regular data science, where we do statistical QC, catching outliers and errors. The gold standard here is, especially when you're looking at something like post-hoc analysis of a clinical trial, can you replicate the listed statistics using the extracted data? And this is a very powerful way to make sure that your extraction was successful without having to check every single record. If the data set says 59.1 of the patients in this trial were female, you can look at your extracted data, calculate the percentage of patients that are female according to the extracted data, and make sure that those match. If you do this across the outcomes and across different demographic reporting guidelines from the statistical pages that you identified in the triage, then you're able to have pretty high confidence about the quality of your data. Additionally, it is reasonable to do other types of statistical QC, like outlier detection, which will allow you to find situations where you extracted the wrong column or columns merged together, by making sure that you're testing whether the data as a whole makes sense. This is something that our bfLEAP platform is particularly good at, so we count ourselves lucky to be able to use it for a slightly different purpose here, to be able to certify that the data that we're extracting doesn't have anything that looks artifactual at face value. So why does this work? Why is this better than doing one big ChatGPT prompt, or why is this against the current that you might be seeing, where AI-based writing or AI-based analyses tend to not deliver the punch that the hype would let you believe they can? Well, the first part is task decomposition. You need to keep it to something specific. We do one page, one task, clear success criteria. You don't want to be writing prompts where you're trying to extract all patient data from 100 pages. You want to extract adverse events from page 47, use schema. You want your outputs to be structured. You don't want to have some free text from ChatGPT adding to your already huge pile of free text that you need to interpret into a data set. You want to structure your data, not just so that you can make decisions immediately after that, but so that you can fit them into interfaces for human review, and so that you can fit them into the types of structures that data scientists can use to examine how well everything is working. Multi-layer QC is another reason that this works. You want to make sure that you're not just checking whether things are correct at the very end, but rather that you're keeping different gates at different steps. There are many steps in the reasoning required for performing this very large task. And by breaking that into different gates and checkpoints, you can track where are you having problems, where do you need to iterate, what kind of consistency are you seeing versus your expectations, either based on the document or the rest of the field. And finally, to iterate on failures. When we started seeing that handwritten characters were not being extracted well, we expected that this might be the case, and we were ready to exclude some percent of the data set based on it being handwritten or poorly scanned. but iteration on these failures to try to see what we could add to the actual images of the pages to help the language model be more effective in its extraction ended up paying off quite a bit. It helps us to refine fail extractions in general to do this. But as a operations point, it helps you to escalate ambiguous cases. Sometimes the failures are not failures because the language model is bad at it. Sometimes the failures are failures because there's an issue with the data set that should be escalated. The key in general is systematizing many small useful queries and then digest those answers into larger answers. This is something, by the way, that you'll see is prevalent in the agent LLM literature. When you're looking at the task that you're giving an agent, the probability that it is correct is directly proportional to how little time it would take a human to do this. You want to make these really digestible tasks that are really hard to get wrong. What we learned as we did all of this is that verified automation is really the answer. It's not full automation. We're also very surprised by model capabilities. We're able to handle complex layouts, ambiguity. Schema inference in particular was very strong, and the coverage of medical terminology was fantastic. As I mentioned, pre-processing choices were an area in which we learned a lot. There's enough details here that a separate talk would be good to cover them. But your prompt and how you process the images are as important as the source data. Not only, it is part of the whole crap in, crap out principle. We learned that human in the loop is critical and that statistical QC is mandatory. This is not something nice to add at the end. This is something that you need to be doing to catch system errors and make sure that your distribution checks are happening, that you're not just assuming that the data set, the notation, or the language model are perfect translators of the large scale patterns that you're trying to feed into your actual statistical and machine learning analysis. So that ended up being the story end to end with one of our clients. We used bfPREP to go all the way from structuring the trap data, extracting all of the data from these tens of thousands of pages of PDFs, putting it into a common data model. In this case, we are delivering included OMOP tables already populated with all of their data sets, building relevant feature combinations that we could add both from the OMOP tools and from being able to use agents to suggest new combinations that would be useful, and building relevant categorical features so that we could slice and dice these data sets with more reasonable granularity or resolution than what the perspective is when you're writing down the data for logging. And we're very happy to say that this is something that we offer as a service now. It's a pipeline that has grown a lot over the past year and that feeds very, very nicely into our ability to find a pattern in the data. If you're a data scientist, you know that some of the trade-offs of doing this kind of approach might be introducing too many features and having to deal with large dimensionality of features that are correlated. bfLEAP specializes in making sure that you can find the actual features that you need for the task at hand and that you can do unsupervised learning on your data set to understand what are the groups that are emerging here that might be able to help you conduct better clinical trials, might accelerate your target discovery efforts, might help you interpret your high throughput screens where there might be subpopulations you're not aware of, or look at how you can do drug rescue by identifying subpopulations of patients that might have a different effect to the drug being tested than you would be able to notice if you were treating them all as one large population. The whole coverage here is looking at the unsexy 90%, the fact that you need verified automation for real world data work. There's a common saying in the field that most of the effort, 90% of it, is used on feature work when you're trying to do a machine learning pipeline. And that is the reality. And it's not sexy. Feature work and data structuring, they're not flashy. No one's going to put you in a conference abstract or a headline because you got really good at your feature work and you're not going to win awards. But this is what enables ML and statistical analysis. It separates working models from broken ones, and it consumes the most effort out of anything else that you're doing in your pipeline in practice. We are showing you a way to do agents done right. It's systematic use, it's not ad hoc prompts, it's structured outputs with types so that you can understand and process what is coming out of the agents, and so that you can repeat the same task on the same agents and see how much the answer changes if you ask the same question twice. That is hard to quantify if you don't have structured outputs. And finally, human oversight and statistical QC at scale. In order to be able to look at your data as a whole, at your extraction as a whole, and decide when you're ready to move on to analysis and statistics to make decisions about next steps for your drug program, next steps for your clinical trial, or next steps for your analysis. Now, if any of this has resonated with you, if you see some of these problems with the data sets that you're working with now or data sets that you're acquiring, let's talk. My contact information is below. You can email me at juanfelipe.beltran@bullfrogai.com. And additionally, you can go to our bullfrogai.com/bfprep website if you'd like to see what we can do for your data sets. Thank you for joining today. For anyone who has registered to this webinar, we're offering a complimentary data readiness audit. You can let our team take a look at some subset of your data, anonymized or otherwise, and we can show you what we can do with that data set to get you to the next step of analysis. Thank you all for your time. And Ryan, I think this is a good time to open the floor for questions.
Yes, thank you very much for that very insightful presentation. As we move into the Q&A session, I'd like to direct the audience's attention towards the handouts module within the GoToWebinar control panel, where they'll find some additional documentation related to today's presentation to download for some further reading. I'd also like to invite the audience to keep sending their questions or comments right now for this Q&A portion of the webinar. If you're attending via GoToWebinar, please use the questions window. And for those watching on LinkedIn Live events, feel free to post your questions in the comments section. Now, I've already received some questions from the audience, so we'll get ourselves started with those. Getting started here, the first question just wants to know, it says that there are many AI clinical data platforms emerging. They ask, what are the sort of 1 to 2 most important ways that Bullfrog AI's solutions differ technically or commercially? And how do you expect this differentiation to drive customer adoption?
That's a great question. One of the strongest cases that I would make for why we are different is the fact that we are not a data extraction service to begin with. We operate in advanced analytics for complex data sets across fields and across indications. These same tools and the same framework that we use for working with the actual analysis is what motivates the way that we do data extraction. And we have seen that that ends up making a huge difference, not just in the amount of time that it takes, but in the guarantees that we're able to make about quality and about outlier detection, even at the extraction stage. Another thing that I think makes us fairly unique is the way in which we systematize the way that this is happening. There's a lot of data extraction platforms that are able to do data extraction in general for these types of documents. But what we do is make sure that these are things that are not operating in a vacuum, not something that you're using as software as a service, not something that you just have one person kind of overseeing, looking at a couple of KPIs, but something that is being done in concert by domain experts in their fields and data scientists and computational biologists, so that you have many eyes on the way that this is happening and you're keeping relevance in sight to not just extract the data, but supplement it. I haven't seen anyone who is doing this type of supplementation right now.
All right, thank you so much for that answer. The next question that I have here wants to know sort of what, it's a little, sorry, we're going to go on to another question. This one's pretty similar. So, okay, so this one wants to know, how do you ensure clinical validity when AI is proposing disease groups?
Clinical validity is always going to be a difficult decision that's already happening when you're trying to do anything with clinical data sets. What we do is make sure that every decision is logged, not just in its justification, but in what was the overview process for it, who signed off on it, who approved it, and keeping that chain of accountability. This is what allows us to turn a process that's already happening informally, where you might ask a colleague that works in the oncology field or a colleague that works in the neuroscience field to give advice on how you make a clinical decision about a couple of features, and turn that into an actual interface where they're able to make their decision and gets automatically logged and included in the provenance for everything that you're receiving at the end of the day. So that is what we do to try to certify the clinical validity. Additionally, as I mentioned, we make sure that the papers that are being used to make this decision are there, and we take it very seriously to make sure that this is not something that we include as a citation, but something that, as I mentioned, starts the work. We go into the papers, we make sure that this is what you want to do, and that it is either part of regulations or that it is accepted in its field.
Perfect. Thank you so much. Now, the next question here is wondering sort of what is the typical project timeline to launch an AI-enabled workflow? Are we looking at weeks or months?
We're looking at weeks. These tools tend to work very fast and they keep working faster. The first time we ran a full extraction pipeline on a subset of that 10,000 page data set, I think we were looking at about 2,000 pages, and it took us maybe 2 days to run end to end. We ran it again about 3 weeks ago, and it took less than an hour. So the process is accelerating as fast as we can write the code, make it more efficient, and the models themselves are getting faster and more intelligent.
Fantastic. Thank you so much for that answer. Now, next up, someone wants to know, how do you ensure, how do you handle just data security?
Data security is a great question. When we're looking at this type of framework where we're using a language model at any step, I'm just trying to, let me go, yeah, let's say you're using some source IDs that might include patient data, especially if you're doing MCP and giving the model some access to your data set. Immediately there should be flags, alarms going off. Where is this data going? How is that going to be handled? The answer is we handle that by making sure that first that we're compliant with SOC 2, with HIPAA, that we're not sending anything to a service that we do not have a business agreement with. But more explicitly, we can run all of this in the same machine. Open source models now coming out of OpenAI or other parties are able to run in the same machine that you are in, so that you don't have any issues with security being leaked. All the queries can occur within your own silicon. If you are in the type of scenario where you don't even want these systems running on our silicon, we are happy to set up on-prem services for clients to fit their needs.
Fantastic. Good to know. Thank you. The next question is someone very curious about how you can stay current as AI technology evolves very rapidly.
It is very difficult. That is a fantastic question. I think staying current on the evolving AI technologies, one, you need to try them out constantly. When we first got access to things like ChatGPT 3.5 and everyone was frustrated about how poorly it conducted certain tasks, I would still make it a habit to ask some questions that I already had answers to these models, and work with domain experts in my network who ask questions that they found difficult and have them assess whether they like the answer or not. That is the primary way in which I think keeping track of how performance is going can be internalized, because it's not just about statistics. Column 2 is the statistics. You can, if I was going to name one area to look at, I would recommend taking a look at Humanity's last exam. It's a benchmark that asks very difficult questions across fields to every language model that is released. And they are scored not just on how correct they are, but on how often they were correct and confident about their answer. They were incorrect and confident about being correct. Being able to track these kind of metrics is vital. I think those would be my top 2 suggestions.
Fantastic. Again, thank you so much for that answer and really for all of the answers today, but we have reached the end of the Q&A portion for the webinar. Please note that if we couldn't get to your questions, the team at Bullfrog AI will follow up with you, or if you have some further questions, you can direct them to the email address that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as we exit, and your participation is appreciated as it helps us to improve our webinars. Now, additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com so you can register for this event and then gain access to features such as viewing that recording and more. Now, please join me once more in thanking our speaker for his wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you for coming.